Hello, I'd like to start by thanking the organisers for giving me the opportunity to present at the second KDOC Science Online Conference. My name is Professor Andrew Hill. I'm from the Latrobe Institute for Molecular Science, which is based at Latrobe University in Melbourne, Australia. The title of this presentation is Extracellular Vesicles and Diseases of the Aging Brain. The term extracellular vesicle was coined well over a decade ago now, and it's a collective term to describe the various types of phospholipid bilayer enclosed vesicle structures that are released by cells. And these vesicles can range in size from about 30 nanometers in diameter to vesicles that are over a micron in diameter, and they include vesicles such as exosomes, microvesicles, apoptotic bodies, and large oncosomes. The way these vesicles form by the cell can differ depending on the type of vesicle being produced, and they can arise from mechanisms such as the outward blebbing of the plasma membrane or through an endocytic pathway which involves exocytosis of vesicles that form into structures called multivesicular bodies. As we can see from this diagram here, these vesicles not only have different sizes, but they also have quite a lot of heterogeneity, which can arise from the different cargoes that these vesicles can contain, as well as their membrane structure, and as I've mentioned, the size as well. These vesicles are able to pass through biological barriers, which is quite relevant to the diseases we'll talk about today, because they can cross membrane barriers such as the blood-brain barrier. And they can also modify the physiological state of both the cell that releases the EV and also the cell that takes up a particular EV. And again, that's quite relevant when we're talking about disease processes, particularly in the transfer of misfolded proteins, as I'll discuss today. And the kinds of cargo that can be carried by these vesicles can include things such as proteins, which come from the cell of origin, but also nucleic acids, particularly RNAs, and also lipids. And I'll discuss the role of some of the proteins and nucleic acids in these diseases of the aging brain and their relationship with EVs. So there are many diseases of the aging brain, and the diseases I'll focus on today are those known as neurodegenerative diseases. And these are a large group of diseases that affect our aging population. They share some commonalities, and many of these diseases are associated with proteins that change their shape from their normal native conformation into misfolded forms. And these misfolded forms of these proteins can deposit in the brain and form protein plaques, which are quite often formed of amyloid fibrils. And the diseases I'm talking about include things such as Alzheimer's disease, Parkinson's disease, and the prion diseases, which I'll talk about today, but also Huntington's disease and amyotrophic lateral sclerosis or motor neuron disease, which I'll also discuss today. And while these diseases all have quite different symptoms, they're all neurodegenerative, which means you see degenerative processes occurring in the brain. And they're all associated with various different proteins that misfold and form those deposits in various different brain regions in the different diseases. These diseases aren't diseases of the younger age groups. Aging is a major risk factor for these diseases. And so as our population is getting older, more and more people are developing Alzheimer's and Parkinson's disease, which are clearly forms of dementia. And being a disease of the brain, the diagnosis of these can be quite difficult. And it's usually only after clinical symptoms have occurred that it's possible to make a diagnosis as to which particular neurodegenerative disease a patient may be suffering from. Now, my lab's been studying EVs in these diseases for about 15 years now. And part of this is due to the growing evidence that EVs play quite strong roles in the maintenance of the central nervous system. EVs are released by all different neuronal cell types, including oligodendrocytes, microglia, astrocytes, and neurons themselves. And they're involved in a lot of homeostatic processes, but also in supporting stress resilience, synaptic transmission, and neuroprotection. As I mentioned earlier, EVs can cross biological membranes. So EVs can transfer from the brain into the CSF, and also they can cross the blood-brain barrier into the circulation. So EVs from the brain can be found in the circulatory system. And as I'll talk about later in this presentation, this is quite important for using EVs in the circulatory system as potential biomarkers for brain diseases. So a common feature of these neurological diseases is the spread of these misfolded proteins in the brain. And by that, I mean that the pathology starts in one particular brain region, and during the disease process, which can be many years in the case of diseases such as Alzheimer's and Parkinson's disease, you'll see the spread of those misfolded proteins occur through different brain regions. And so we're interested in studying the role of EVs and the role that they play in spreading those misfolded proteins to these different brain regions. So over the last decade, there's been a lot of work showing that some of these major proteins that misfold and deposit in the brains of individuals with these neurodegenerative diseases are found associated with EVs. And these include the proteins such as alpha-synuclein, which can be found in its native monomeric form, and also its disease-associated aggregated form associated with EVs. Also proteins involved in amyotrophic lateral sclerosis or motor neuron disease, which includes superoxide dismutase 1, FUS and TDP43 can also be found in their normal and disease-associated forms associated with EVs. Also for Alzheimer's disease, both tau and amyloid beta and its precursor, the amyloid precursor protein, can also be found associated with EVs. And finally, in terms of prion disease, the normal form of the prion protein, PRPC, which I'll talk about in a moment, and the abnormal form PRPSC can both be associated with EVs. So this provides some evidence that because these EVs can contain both the normal and the abnormal disease-associated form of these proteins, they can represent a pathogenic mechanism for spreading these misfolded proteins throughout the brain, and also maybe present a diagnostic or therapeutic target to either help diagnose the disease by detecting these misfolded proteins, or in terms of trying to reduce the number of EVs carrying these misfolded proteins, that can be a potential therapeutic target, which is something we'll touch on as well. Another cargo, besides those protein cargoes that EVs carry, EVs also carry nucleic acid cargoes, and I'll be talking particularly around microRNA cargoes and their role as potential biomarkers for these neurological diseases in this presentation. So our work started on looking at quite a rare neurological disease called prion disease, which is known as Creutzfeldt-Jakob disease, CJD, in humans. And you may have heard of it occurring in cattle, where it's known as bovine spongiform encephalopathy or mad cow disease. This is a protein misfolding disease where a normal cellular protein, which is called PRPC, the cellular prion protein, and here's a molecular dynamic simulation of the protein. Here it has 3 alpha helical regions here, and it has a long N-terminal domain that is relatively unstructured, and it's tethered to the cell surface via a GPI anchor. In the disease process, these alpha helical regions change into beta sheets, and the structure of the protein changes. It becomes insoluble and forms amyloid. And this beta sheet-rich structure changes its protein and induces this conformational change in further copies of the normal prion protein. And so this disease-associated misfolded form of the prion protein is known as PRPSC. And we've been interested in looking at how this abnormal form of the prion protein can form within cells and how that can spread between cells, be it neurons or other cells, and induce the further conversion of this normal prion protein into the disease-associated form. And there are several mechanisms by which this can occur. One is where you have the abnormal form of the prion protein, shown in red here, on the outside of the cell. And during cell-to-cell contact, you can get conversion of the normal form of the prion protein, shown in blue here, on one cell occurring when the 2 cells are in close proximity. Another mechanism by which the normal form of the prion protein can exchange and change its conformation into the disease-associated form is through structures called tunneling nanotubes. And this is where 2 cells, they're not necessarily touching, but they're in close proximity. And one cell can send out a protrusion, which is known as a tunneling nanotube, and the cell can share its contents, including some small EVs. And so this is how this disease-associated form of the prion protein can transfer from one cell to another. The mechanism that we've been focusing mainly on, though, is the role of EVs, particularly those formed through the endocytic pathway, the classical exosomal small EV type of vesicle. And this is where, because the normal and the disease-associated form of the prion protein are on the outside of the cell, when they undergo this endocytosis pathway, which results in the formation of multivesicular bodies, which are these small intraluminal vesicles that contain the normal and the abnormal form of the prion protein. When this multivesicular body fuses with the plasma membrane, it releases its contents, and you have all of these small EVs containing the normal or the disease-associated form of the prion protein, which can then travel to another cell and be uptaken and cause the disease process. So we've been studying this in various different models. We use cell-based models where we isolate the EVs and we look at how the EVs can transfer and induce that protein misfolding. So our model uses an infected mouse brain homogenate. So this is a mouse that's had an induced form of prion disease. And we take the brain homogenate from that mouse, which contains a lot of that misfolded prion protein, and we expose cultured cells to some of this brain homogenate and passage the cells for several weeks to remove the original inoculum. And what we see after several weeks is we see the infection, the prion infection, which is that changing of the normal to the misfolded form of the prion protein occurring amongst the cells. And we use quite a simple biochemical assay to look for the presence of PRPSC, that disease-associated form of the prion protein. We use a protease assay where we can see that in mock-infected cells, which have had no infectious prion protein contained within the inoculum, that before treatment with protease, we can detect the normal form of the prion protein, and after treatment with protease, we see no signal. In cells that have been exposed to prions from this infected mouse brain homogenate, we can see that after the proteinase treatment, the presence of these 3 protease-resistant bands, and this is showing us the presence of that protease-resistant beta sheet-rich form of the prion protein that induces the conformational change. So some of the first experiments we did was to take cell lysates and also small EVs from these infected cells and inject them into a mouse model of prion disease, and we found that the exosomes from 2 different cell lines infected with prions were able to induce a clinical disease in these mice with a very similar time, incubation time, to when we infected just the total cell lysate. So this showed us that the EVs from these prion-infected cells were infectious and could induce a clinical disease in these animals. To look at whether we needed cell-to-cell contact to transfer this infectious prion protein between cells, we did a transwell experiment where we cultured some prion-infected cells on a membrane with a 0.4 micron pore size, and we looked to see whether the infection could transfer across this membrane. And we can see here in the cell blot assay, which is just another way of looking for that protease-resistant prion protein here, we can see that when we have prion-infected cells on this membrane where the cells aren't in close contact, we can detect the infection in these reporter cells at the bottom of the dish. And finally, another experiment we did was to take the culture media from these prion-infected cells. We added that onto uninfected cells, showed that the cells could take up the infection. When we isolated the EVs from this culture media, we could see they were very efficient at transferring the infection. And when we looked at the EV-depleted culture media, we could see that the amount of infection was greatly reduced. So collectively, this is telling us that EVs are a very efficient way of transferring this transmissible misfolded protein between cells. We then looked at the relationship between the release of these EVs and the transmission of this infectious misfolded prion protein between cells. And we did this using 2 compounds that are known to either increase or decrease the amount of EV release. So firstly, we looked at a compound called GW4869, which is a neutral sphingomyelase inhibitor that's been shown to reduce the number of EVs released by cells. And when we treat our prion-infected cells with this inhibitor, we can see we get about a 50% reduction in the number of EVs released by those cells. And when we do that same transwell assay, where we culture the prion-infected cells, we treat them with the inhibitor, and look for the infection in the reporter cells in the well underneath, we can see that compared to the vehicle only, when we've treated the cells with the inhibitor, we see a vast reduction in the amount of infectivity. So this is telling us if we reduce the amount of EVs released by these prion-infected cells, we can slow down the infection transferring between cells. We also used a compound called monensin, which in a dose-dependent manner increases the number of EVs released by cells. And you can see here in the same assay, this transwell assay, as we increase the amount of monensin, we get more infection occurring in the reporter cell line. So this is telling us, again, if we increase the number of EVs released by these prion-infected cells, we increase the amount of infection that's taken up by those reporter cell lines. So while these compounds are somewhat blunt tools of looking at increasing and decreasing EV release, it does show that manipulating the number of EVs released by the cells has a direct relationship to the amount of infectivity that can be taken up, again, indicating EVs are a very efficient way at spreading these misfolded proteins. We've then done further biochemical analysis on the EVs from these prion-infected cells to just look at what is it about the EV and having this misfolded protein makes it so infectious. So we did a series of titrations where we titrated out the number of EVs added to the uninfected cells, the number of prion-infected EVs added to these cells. And so when you see the graph moving towards the left, that means that there's an increasing amount of infectivity as we have, as we dilute the sample, we're still detecting more infectivity. So our first experiment was to compare the amounts of that protease-resistant form of the prion protein in the total cell lysate and with that found in the EVs. And we normalized this on a Western blot, so you can see the cell lysate here. We have the infectious prion protein amount here. And in the exosomes, we've normalized it. So we've got the same amount of the infectious misfolded prion protein in the exosomes as well as the cell lysate. And we can see here in our dose titration that the red line here, the exosomes, are more infectious because the graph's moving to the left than the cell lysate. So this is telling us that with the same amount of the infectious prion protein, the EVs are more infectious than the total cell extracts, and that this increased amount of infectivity isn't due to there being any more PRPSC, that misfolded form of the prion protein, in the EVs because the levels are virtually the same. We then asked the question, is an EV membrane component important for enhancing the efficiency of this infection? So we broke open the EVs using both hypertonic shock and sonication. And here we used electron microscopy just to monitor that treatment to make sure we were breaking open the EVs and maintaining some membrane structures there. So here you can see the control whole EVs before the sonication and hypertonic shock. And here you can see them afterwards. And you can see we've broken open all of these EVs. And when we do that titration curve, we don't see any significant difference between the control, the whole EVs containing that misfolded prion protein, and the ones that had been sonicated and treated with hypertonic shock. So this perhaps tells us that the infection efficiency of these misfolded prion-containing EVs is enhanced by a membrane component, because you can see you've got membrane fragments here which contain some of that protein. We also looked at whether the surface proteins on the EVs played a role in the uptake of the infection. And what we did was we took the EVs from the prion-infected cells, we treated them with proteinase K, so we were shaving off all of the surface proteins from the EVs, and you can see that that hasn't changed the structure of the EVs using transmission electron microscopy. And again, this showed no difference on the dose titration curve. So there's nothing to do with the protein corona on these EVs that's affecting the uptake of the misfolded prion protein. But we saw a major difference when we chemically altered the structure of the EVs by treating with a methanol and a detergent called Sarcosil. And here we can see control EVs, which had this is the line for the control EVs shown in red here. And we can see when we treat the EVs with methanol, we're breaking up the structure of the EVs and the membrane structure. And we can see we get a massive reduction in the amount of infectivity shown here because the graph is now moving to the right. And when we add sarcosils, so we're actually solubilizing those membrane fractions, we virtually lost all of the infectivity. So this is telling us that it's a combination of the misfolded protein in a membrane environment in the EV that's important for maintaining its ability to perform that transmissible action, that infectious action of transferring from one cell to another. This is something that we've also recently seen, and we published this in the Journal of Extracellular Vesicles in 2020, with another protein that's involved in neurodegeneration. And this is the protein called alpha-synuclein, which misfolds and deposits in the brains of individuals with Parkinson's disease. So we took small EVs from a neuronal cell line called GT17 cells, and we looked to see whether these EVs could help accelerate alpha-synuclein misfolding. And this was again to test the premise that EVs might be involved in the pathogenesis of Parkinson's disease. So alpha-synuclein is a protein of about 140 amino acids long. It's natively unstructured, but it does misfold and form amyloid-like structures which cause Parkinson's disease. And it does this through this fairly typical pathway where monomers of alpha-synuclein start to come together to form these oligomeric structures, which then start to aggregate, forming these pre-amyloid structures known as protofibrils, which then form amyloid fibrils. And so this is a classical protein misfolding disease involving the protein alpha-synuclein. We used an assay using a compound called thioflavin T, which is a fluorescent compound where you see increased fluorescence in the presence of these amyloid fibrils. And so here we used it to measure the aggregation of alpha-synuclein, so just the native protein by itself. And if you follow the blue line, you can see over a 24-hour period, we see an increase in alpha-synuclein thioflavin T fluorescence. When we add small EVs to the alpha-synuclein, we can see that this process is rapidly increased. So we're seeing an increase and we're reaching a plateau of aggregation a lot earlier than just the protein by itself. And similar to the experiments we did with the prion protein, if we add a cell extract, we're actually slowing down the aggregation process. So here it looks like the small EVs are accelerating the aggregation process of alpha-synuclein. We also looked at this biochemically using an assay called the protein misfolding amplification assay. And you can see alpha-synuclein by itself, it's about 14 kilodaltons in size. And after 24 hours using this PMCA assay, we can see we get aggregation of the protein shown by these much larger structures at the top of the gel. When we add small EVs to the alpha-synuclein, we can see we get a lot more aggregation. And this is significantly higher than that seen when we add a cell extract, which looks virtually the same as the protein by itself. So this biochemically is also suggesting that small EVs can help alpha-synuclein down this aggregation disease-associated pathway. We also asked a similar question to the prion study as to whether an intact EV membrane is important for enhancing this aggregation property of alpha-synuclein. So to probe the relevance of the membrane architecture, we isolated EVs and chemically modified them again using methanol or methanol and sarcosyl, sarcosyl being that detergent. And here you can see the control EVs here. You can see them treated with methanol and the methanol sarcosyl treatment. And again, it's broken open and solubilized the membrane structures from the EVs. We can see using that thioflavin T assay that the untreated EVs gave a much higher thioflavin T signal, indicating aggregation of the alpha-synuclein protein. And when we treated the EVs either with methanol or methanol and sarcosyl, we greatly reduced the amount of aggregation. So again, this is showing that perturbing the membrane structure and solubilizing the membranes inhibits that aggregation pathway, similar to what we see with the prion protein. So this is telling us when two different proteins involved in two different neurodegenerative diseases of the aging brain, that EV membranes provide an environment that supports and enhances the misfolding of these pathogenic proteins associated with these diseases. So now I'm going to talk a bit about some of the RNA content of the EVs and how they might be used for diagnosing neurological diseases. And so EVs over the last 2 decades have been implicated in a number of different disease settings, and these include cancer, neurodegenerative and other neurological diseases I'm talking about today, cardiovascular disease, and also infectious disease. And because they're associated with these diseases, we can use them as sources of biomarkers by looking at the different cargoes and seeing how the cargo differs between a control or a normal individual and perhaps someone that has one of these disorders. And the different cargoes we can look at include the nucleic acid cargo, proteins, and also lipids. At the moment, there are some technical barriers to doing this and rolling these out as diagnostic tests, and these include methodologies to isolate EVs and also the sensitivity of some of the downstream assays. But there's a lot of work that's been happening over the last several years involved in standardizing these methodologies and validating biomarkers, and this is a really important step in taking these kind of technologies into the clinic. So our first study on looking at does the cargo of an EV change between a normal cell and one that's associated with disease, we performed on our prion cell culture model. And here we took small EVs from control and prion-infected cells and performed small RNA deep sequencing to compare the microRNA profile between control and prion-infected EVs. And in this first study, we identified a panel of 9 microRNAs that were differentially expressed between the prion-infected cells and the control cells. And so we identified these using small RNA deep sequencing, and we validated it using a couple of different qPCR techniques. So this study, which we published nearly 10 years ago, led us to start asking the question, can we use the microRNA profiles of EVs to diagnose neurodegenerative diseases? So our next study, which we published in 2015, was a study looking at serum-derived EVs in Alzheimer's disease. And we took 23 healthy control samples and 23 Alzheimer's disease blood samples, and we isolated the EVs and performed deep sequencing on those and looked at the microRNA profiles between the healthy controls and the Alzheimer's cases. And we found a panel of 16 microRNAs with differential expression between the healthy controls and the Alzheimer's cases. So this was our discovery sample set. We then validated these markers using a blind study where we had 50 samples that we analyzed blind to diagnosis. And all of the patients in this sample set have been diagnosed by standardized criteria, and they'd all had PET imaging for the amyloid beta marker as well. And we found that we were able to correctly identify 13 out of the 15 Alzheimer's cases in this blind set of samples by looking at the microRNA profile in the EVs with these 16 markers we'd identified in our discovery phase. So this test had a sensitivity of 87% by correctly diagnosing 13 out of the 15 Alzheimer's patients. For the healthy controls, we correctly diagnosed 27 out of 35, which gives us a specificity of 77%. So this isn't too bad for an early diagnostic test for a brain disease such as Alzheimer's. And by looking at the PET imaging, we found that quite a few of the patients we misdiagnosed had high levels of amyloid beta. And so by looking at the longitudinal analysis of these cases, if they turn out to having Alzheimer's disease, we think that this will take our specificity to over 90%. And so we're currently wrapping up a longitudinal study at the moment that we hope to publish later this year. So this work was published a few years ago now in Molecular Psychiatry, but it demonstrates that serum-derived EVs have diagnostic value in diagnosing Alzheimer's disease. To really look at how we can try and improve the accuracy of these EV microRNA biomarkers in the periphery, we then started to look at what are the EVs like in the brain of individuals with some of these diseases. Laura Vella, who has her own laboratory at the Florey Institute in Melbourne, collaborated with our lab, and we developed a technique for isolating EVs And at the time, we thought they met the criteria of being termed exosomes from frozen human brain specimens. And so in this study, Laura developed a protocol for isolating EVs from the frozen brain tissue, and it involved fractionation across the triple sucrose cushion. And this work was published a few years ago, again in the Journal of Extracellular Vesicles. And this triple sucrose cushion, it's a density gradient, allows the separation of different size vesicles, with the vesicles found in fraction 2 representing the small EV population. And so commonly when we do the analysis, we compare fractions 2 and 3 because fraction 3 contains some slightly larger sized vesicles as well. And the vesicles we can isolate from these frozen human brain samples are absent for a number of non-exosome, non-small EV markers, and they light up positive, particularly in fraction 2 for well-known small EV markers. So by developing this technique, it's allowed us to ask several more questions, such as, can we identify brain-specific microRNAs associated with disease in these brain-derived EVs? Can we try and capture brain-derived EVs in blood by looking for markers on these EVs that might allow their capture in the periphery? And can we identify preclinical EV microRNA signatures that have diagnostic capacity? So some of the early work we did using this protocol was to look at the protein profile of these EVs. I won't dwell on this too much here, but we found, and this is again in the publication from 2017, that during a shotgun proteomics approach on the brain tissue-derived EVs, that they contain many of the proteins I spoke about that are involved in these different neurodegenerative diseases, and they contain the top 25 proteins found in small EVs. So this protocol was, this was helping to support the notion that this protocol was actually isolating EVs from the brain tissue that weren't contaminated by other cellular material. We've then done a lot of work on comparing the profile, the EV microRNA profile from the brain-derived EVs with those found in the periphery. And we published this in 2020. And the aim was to identify brain-specific AD biomarkers that we could then detect in the blood. And we did a number of different bioinformatic comparisons, comparing the brain tissue and the brain EV microRNA profiles, and comparing them with those in the serum. And we performed various different comparisons. I don't have time to go into them today. But what we did find was that the RNA species that we could detect using the next generation sequencing in the brain-derived EVs and the total brain tissue, and we also compared that larger EV fraction in fraction 3. And we found that the normal control and the Alzheimer's brain-derived EV RNA profiles were quite similar to those of the brain homogenates. And you can see we can detect a wide variety of different small non-coding RNA species here. And of note here is that the brain-derived EV small EV population, the microRNA and other small RNA profiles is quite different to that we found in fraction 3, again supporting the idea that this triple sucrose gradient is separating different EV populations. And again, this was published in 2020 if you're interested. So we looked at comparing, one of the comparisons we did was comparing the brain-derived EVs from Alzheimer's and normal controls from both the brain and also what we could see in the serum. And we saw some interesting correlations here. We found this graph here where we're comparing the serum EVs of the healthy control versus the AD samples. And we've plotted this on the x-axis against the brain-derived EVs for the Alzheimer's and the normal controls. And we can see a number of microRNAs that we see differentially expressed upwards in both the brain-derived EVs and the serum, shown in green. And shown in green down here, we see a population of microRNAs that are downregulated in both the EVs from the serum and the brain-derived EVs. But we also saw a number of microRNAs where we saw an inverse correlation. And this is where they can be down in the brain and up in the periphery, or up in the periphery and down in the brain. So we think there's some really interesting biology going on here where we're detecting certain things in the blood that are representing what's happening in the brain, but some of those things that are either up or down in the brain are doing the opposite things in the periphery. So we're looking at that at the moment. And again, this data was published a couple of years ago. And this is just to show that we've seen this in another disease, in Parkinson's disease, where we've done a similar analysis of comparing brain-derived EVs from healthy controls in Parkinson's patients and comparing them with the serum-derived EVs from healthy controls in Parkinson's patients. And we can see a number of microRNAs with the same expression patterns, up in the brain, up in the serum, down in the brain, down in the serum, but also in red, we see populations of EVs with that inverse correlation. Another disease that we've started to look at is amyotrophic lateral sclerosis, or motor neuron disease. And here we can show that we've been able to also isolate EVs from motor cortex tissue taken from patients who've died of ALS. And here we can show the electron microscopy of the EVs we can isolate from the frozen brain tissue. We see the size profile here using nanoparticle tracking analysis, and also the presence and absence of known small EV markers in the fraction 2 from the gradient. So we've been able to classify the EVs from fraction 2 as being small EVs from these ALS brain tissue samples. We did a proteomic analysis of these samples, and we found 16 differentially expressed proteins compared with the healthy control and the ALS samples. And using this unbiased screen of the brain-derived EV protein profiles between the healthy controls and the ALS samples, showed us that there are these 16 proteins, and many of them have been known to have direct links. The ones with arrows here have been shown previously to have direct links to ALS. So we thought this is really interesting, that just analyzing the EVs, which represent a subpopulation of the cell, we're actually highlighting pathways that are well known in the pathogenesis of ALS. And we saw some overlap between the small EV biogenesis pathways and macro-autophagy, and also that packaging of stress granule proteins into the ALS brain-derived EVs might compensate for lysosomal dysfunction, which is caused by the accumulation of these stress granules, which is a key feature of ALS. And this is work that, again, that we published in 2020, and we wrote a review for the Journal of Cell Biology on this as well. We've also started to look at the brain-derived EV microRNA biomarkers in these ALS samples. And our goal is to try and build up a dementia panel where we have a panel of microRNAs that can discriminate between these different common neurodegenerative diseases. And so far, we found 4 quite significantly differentially expressed microRNAs in the brain-derived EVs from the ALS samples. And we're working through these at the moment. So this is currently unpublished data. So just coming back to wrap some of this biomarker study up, we performed a long-term study on looking at the role of EVs in prion disease. And we set up some years ago a long-term study on animals infected with prions to look at the longitudinal analysis of these EV microRNA biomarkers to ask the question, do we think we can diagnose someone on the cusp of developing clinical symptoms? So we've taken this mouse model where we inject mice with prions, and they have a very characteristic incubation period where they develop disease at about 13 weeks after they've been inoculated with prions, and they suffer terminal stages of the disease about 20 weeks after they've been inoculated. And we did a series of timed culls of these animals, and I'm really going to talk about 3 of these time periods, one being 3 weeks after inoculation, where we see no pathology in the brain, and we see the presence of none of that protease-resistant prion protein in the brain of the infected mouse. At week 13, we're starting to see some clinical signs, and we're starting to see some mild pathology. So you can see start to see some vacuolation occurring in the brain tissue of this infected animal here compared to an uninfected control. And we can start to see the presence of the protease-resistant prion protein occurring here. And then we looked at the terminal stage of disease, which is about 20 weeks after inoculation. And here you can see quite widespread vacuolation in the brain compared to the control here. And you can see when we do the biochemical analysis of the brain tissue, the brain is loaded with this protease-resistant form of the prion protein. So at week 3, we're looking at a very preclinical phase. Week 13, we're looking between this preclinical and clinical phase, and terminal is the very end stage of disease. And this work we published last year in Communications Biology. So we did various analyses of tissues from these animals. We looked at the brain tissue, we looked at the thalamus, which is the brain region which is most affected in this mouse model, but we also looked at the serum of these mice. And we took the EVs out of the serum of these mice at those 3 different time points. And we looked at the EV microRNA profiles at those different time points and compared them between the infected and the non-infected mice. And what we found is that as early as 3 weeks, we could see some microRNAs that were upregulated in the infected animals, that then at week 13, it's shown in the middle here, started to become downregulated, and then in the terminal stages, they were downregulated compared to the control sample. So they start off upregulated, still upregulated at week 13, and then downregulated in the terminal stages of disease. And as we compare the week 3 with week 13, we can see different patterns occurring where we can see some microRNAs that are really upregulated. If you look at the middle bar here, at that middle stage of disease, and then they're downregulated either in the preclinical or the terminal stages. And of course, in the terminal stage of disease, we can see some microRNAs that are quite upregulated. So from this, we're able to build up panels of EV-associated microRNAs that could represent the different stages of the disease. And when we looked at the pathways of those microRNAs, so the targets of those microRNAs, they all tended to be associated with known pathways involved in prion disease, such as protein processing in the ER, prion disease itself, endocytosis, and also axon guidance. And so this is quite important when you think about we're taking this out of the serum of these animals, from the EVs from the serum of these animals, and not from the brain, and we're picking up these well-known pathways. And from that study, so there's a lot more work in the paper where we showed the microRNAs from the animals. We actually built up a panel of 14 microRNAs that we think can diagnose prion disease from healthy controls. And in a collaboration with the National Reference Center for Prion Disease in Germany, we looked at some human samples. So these are very rare samples. We were able to isolate EVs from serum samples from some healthy controls and then some individuals who died from Creutzfeldt-Jakob disease. And using our panel of 14 EV microRNAs, we're able to get quite good sensitivity and specificity of 86% and 89% respectively for this blood test for detecting prion disease in these cases. So this again shows the usefulness of microRNA EV biomarkers, and also that there's some translation between the different systems from our cell models to the animal models and to looking in our human samples as well. So just to conclude, I've shown you that the structure of whole intact EVs is important for ensuring the efficient transmission of prion infectivity and in promoting alpha-synuclein misfolding. So disrupting those membrane structures affects the way that the transmissibility of these protein misfolding parameters occurs. Also, that analyzing the RNA from EVs from prion-infected cells, the brain and the serum of mouse models reveals specific signatures that can be used as biomarkers to diagnose this disease. And that these microRNAs we're finding in the EVs have links with disease pathways. Also, I've shown you that brain tissue-derived EVs offer an opportunity to interrogate pathological processes that associate with these neurodegenerative diseases that involve EVs. We know that EVs are involved in the transfer of these misfolded proteins, but also that they contain biomarkers that we can use in the diagnostics. So they're involved in the pathogenesis as well as potential uses in diagnostics and potentially therapeutics. And then I've also shown you that the EV microRNA signatures are associated with Alzheimer's and prion diseases, also less so in Parkinson's and ALS using clinical samples. So with that, I'd like to thank you for your attention, but also acknowledge the many people who've been involved in this work over several years. I particularly like to acknowledge Leslie Cheng, who has led the EV microRNA project for the last 10 years, and also long-term collaborator Vicky Lawson for collaborating on the prion animal studies, and all of our all the members of the lab, past and present, collaborators at the Florey Institute, our University of Melbourne, our international collaborators, and also our funders. And thank you for listening, and I look forward to taking any questions that you might have. Thank you very much.
Thank you. We just had another wonderful presentation, and Professor Andrew Hill is in. So we have a couple of questions. So the first question is, was there a difference in amount of exosome produced by between prion-infected cells and normal cells?
So that's a question we haven't formally answered yet. Part of that is to do with biosafety reasons. When we're working with our prion-infected cells, we can't put some of that material in the instruments that we use for counting the EVs.
I see.
So we are working on that at the moment, because I think that is a really important question as to whether the cells that are producing this transmissible protein are in fact producing more EVs, whether it affects the pathways involved. So that's a great question and something we'd really like to find the answer to.
The second question from the audience, why or how does EV membrane stimulate protein misfolding?
So membrane lipids and membranes can promote protein misfolding. Alpha-synuclein is a really good example of that, because alpha-synuclein natively actually has little structure, little tertiary structure. But when it comes into contact with lipids, it actually gains some structure, and in the right environment, the structure can be those beta sheet-rich forms that form aggregates and amyloids. And I think in the experiments I showed today, where we tried to break down the EVs to see what left that prion protein being infectious, it really did need that lipid environment, and whether it's really a negatively charged cofactor that's involved, or it's really the lipid itself, still remains to be determined. There's been, in the prion field, a lot of studies, many carried out in Korea too, where we've tried to take misfolded prion protein, add it to negatively charged molecules to gain those infectious properties, and nucleic acids have been shown in some cases to be able to do that. So maybe the lipids are sort of helping to stabilize those misfolded forms of these proteins.
Okay. I appreciate your in-depth research on exosome in neurodegenerative disease. Are there any current exosome-based therapeutics for neurodegenerative?
That's another great question, and I guess you could look at that from two directions. One is whether by modifying the number of extracellular vesicles released in the brain, that might be one way of slowing down the disease process. For instance, if you were to slow down the release of EVs containing amyloid beta or the misfolded prion protein, could you slow down the disease process? And there have been a number of animal studies that have shown that compounds that reduce the number of EVs released, and these are compounds such as the neutral sphingomyelinase inhibitors, can slow down the Alzheimer's process in mice. I guess in humans, we need to ask the question, if we slow down the EV release, are we interfering with the normal function that these EVs might have? So might there be effects we don't yet know about in doing that? So I think that's an interesting question. The other way of perhaps using the EVs is to maybe load them with therapeutic cargo. There are many different ways of loading EVs with therapeutic cargoes. They could be nucleic acid based, such as some of the microRNAs that go up or down. You could maybe load or not load certain microRNAs into the EVs, or proteins as well. And so that might be another way of using EVs to therapeutically treat some of these neurological disorders. And there has been some work showing that can be done as well. And I think you're going to hear about that in the talks later on today as well.
Here's another question. There are lots of exosomes in the blood. How you can discriminate exosomes derived from the brain from other exosomes of different origin?
Sure. So that's correct. There's lots of exosomes and EVs in the blood, and they're going to come from many different sources within the body. You know, the question is how many of these that you find in the blood might come from the brain. And so the work I've shown you today is where we've just taken all of the EVs from the blood and looked to see, are there signatures that might have some diagnostic potential. So we haven't tried to look specifically for brain-derived EVs in the blood. There are some studies looking at that using some molecules to pull down what are purported to be brain-derived, neuronal-derived, or astrocyte-derived EVs in the blood. But our approach has been to look at everything just to see if we can get that diagnostic without having to go to those fractionation techniques, those isolation techniques. And also, that's why we also wanted to look in the brain itself to see how does the blood profile compare with that of the brain. So I believe that we are able to get a good diagnostic signature looking at all of the EVs in the blood without having to specifically look for brain-derived EVs. You might get a better signal to noise ratio by going for the brain-derived EVs in the blood, but at the moment we haven't really felt the need to go down that route.
Is there any possibility that tau protein is also associated with exosomes?
Sorry, I didn't quite catch that question.
The tau protein is also associated with exosome?
Yes, it is.
Yeah.
So the tau protein, which is also involved in Alzheimer's disease, is also misfolded forms of tau, and also the hyperphosphorylated forms of tau are associated with EVs. So I think it's a really interesting question why all these neurodegenerative proteins do share that common mechanism of being associated with EVs. So yes, tau is certainly another protein that is associated with EVs.
Okay. I also have one question. The exosome is originally regarded as a garbage can. So it is not surprised to see many exosomes from infected brain cells release many exosomes which contain the misfolded protein. So once it is released, and it is, as you mentioned, it is internalized by the recipient cells, what would be the fate of internalized exosome in the recipient cell? Do you see any difference between the normal exosome, the exosomes from the normal cell, or exosomes from the infected cells?
We don't see much difference in the uptake between the normal and the infected cells. But that's a really good point too about, you know, the function of exosomes potentially as being these entities that take out the trash, take out the misfolded proteins that the cell no longer needs. And a common feature of all of these diseases is that they affect people in our aging populations. So they're not diseases of childhood or early to middle age. It's later in life we get these diseases. And it might be that early on in life, exosomes taking out these misfolded proteins, which can happen by chance, is an effective way of removing them from the body. But as we get older our cells might lose that capacity to put out the trash. And so that could be one potential mechanism where aging comes into how cells process. And we do see in a number of models a breakdown in, proteostasis as we get older. So yeah, it could be that function is really important, and we lose that as we age, and that's when these things can start to go wrong.
We have a couple of more questions from the audience. Could you elaborate on the benefits of using exosomes on diagnostics over other biomarker?
So, I mean, we find that using exosomally derived or EV-derived microRNAs gives a very robust signal in our assays. And because the EVs protect their contents, so we find that by looking for the nucleic acids contained within preparations of EVs, that they're protected from nucleases which exist in the extracellular space. So we think that we get a much better signal to noise by looking for EV-associated RNAs, rather than looking at just total RNAs in the blood without any EV isolation. So we think it gives a much more reliable signal.
There's one more question here. Do you think eliminating infected extracellular vesicles from the blood can mitigate the disease?
That might be one way of mitigating the disease. I think how many of them come out of the brain into the blood, I think also, we need to think about that too. But quite how we'd remove them, whether you needed some kind of dialysis type system, we need to think about. But yes, it could be one way of doing that. I guess thinking about all those other methods of reducing the number of EVs released, etc. So
we have Pan is online, in Joy also in. So you two guys have questions, please go ahead. No?
Thank you so much. No questions from me.
Yeah.
Fantastic presentation.
Well, maybe I'll ask one. Andy, what about the other markers that you might be able to find in these EVs, that might be higher occupancy or more diverse? Where do you think we're going to move in terms of the kinds of nucleic acids that we look at? Will it continue to be microRNAs always, or might there be other ones?
Oh, look, I mean, that's a great question, Ken. And I think given there's a whole suite of small non-coding RNAs that you can isolate from these EVs, I think looking at microRNAs is a bit easier because we have the right bioinformatic tools. But long non-coding RNAs plus all those other small non-coding RNAs, I think that there could be some other really nice markers in there. But I think it's having the right tools to do that analysis that which I'm sure are going to come. So for those researchers that have been doing a lot of sequencing, they're going to have lots of data that they can go and mine when we can sort of look at these other markers as well. And I guess it also might be true for maybe proteins, and even looking at post-translational modifications on proteins would be another good marker to look at. And more people now are looking at the actual lipid content, which can differ between normal and Alzheimer's, for instance, EVs. So I think there's a bit of a gold mine in these little packages, and it's really just about having the right tools, the right sensitive tools. Looking at clinical samples is always challenging, but I think we're getting much more sensitive assays now, and so I think microRNAs are probably just the start of the things that we could look at, and maybe a multi-parametric test might be where we end up to get the most sensitivity.
Good stuff. Thank you, Andy.
Thanks, Ken.
If we don't have further questions, it's time to close the morning session. So Andrew and Ken, let's very thankful for joining this conference. Thank you.
My pleasure. Thank you.
Yeah.